So these are our stated goals. We'll take a look at the anatomy of blood vessels. We'll make sure that you know the difference in the arrangement of veins versus arteries. We'll go about starting the process of memorizing all the different arteries and veins in the fetal pig, being able to identify those. That's what you'll be probing around today. And then we'll talk about three major differences between the fetal blood system and the adult blood system. Think now, there really is a functional difference between being a fetus and being a living, breathing, born piglet. One of them is getting their oxygen from their lungs, one of them is getting their oxygen from their belly. One of them does not have functional lungs. One of them is getting their nutrition from outside of their belly, one of them is getting them from their belly. So we need to know those differences because it matters. How do you keep a fetal brain going? Getting enough oxygen. So I laid this out in greater detail. The test will look like, again, you'll need to know the anatomy of the blood vessels. You'll need to be able to trace the flow of blood through all vessels, just like we do air, just like we do food that you're eating, just like we'll do light coming through the eye and everything else for the rest of the semester. Know the whole pathway, trace a drop of blood through the vessels. You'll need to know the functional difference, that is, what vessels are carrying rich oxygenated blood, rich nutrient blood, and how does that change in the fetus versus the adult. And I already warned you to take a look at lots of internet images, take a look at different pigs in the lab, and see if you can identify the same structures. Again, that's listed there. And as a practical matter, when you're dissecting today, you can take the lungs out pretty handily to start getting a look at the back end of the heart. I think it's going to be important to keep that intact so that you can see the foramen ovale, not the ductus venosus, but the Ductus Arteriosus. Those are the fetal structures. And eventually you're going to take out the various viscera today, take out the guts essentially. But before you do that, try to find the actual blood vessels that are servicing those structures, because it may not be clearer once everything's out. So practical notes. So blood vessels have, both arteries and veins have a common morphology. They have a layer of connective tissue on the outside. There's a basal lamina here in between that and a layer of muscle. In between that there's another basal lamina that services this interior epithelium, which is called the endothelium. So I think we kicked around this term before. Remember, endothelium is the specialized term for these very flat squamous epithelium on the inside of all blood vessels. You will be forced to tell the difference between an artery and a vein based only on their appearance. So, what is the big difference between an artery and a vein functionally? Contributions from the class? Pressure, yes, absolutely. Pressure because arteries do what versus veins doing what? Right, arteries push the blood through the system and veins are the drains. They bring the blood back. So the veins require less pressure, therefore, as a structure, they can be a lot floppier. The arteries, however, have to take all that pressure from the heart beating, so they need a lot of support. And that support comes in the form of a very thick layer of muscle. You know, they have the same layers as just described, but the layer of muscle is much, much thicker. And that is the only difference that you'll really need to be able to pick out between the two structures. Michael?
Other than, like, regulating the amount of blood that goes to, like, an extremity, we're talking about blood vessel constriction, why else do blood vessels need smooth muscle? I mean, because the heart is moving from here. So, is that really it?
My understanding is almost purely structural, but the blood vessels can regulate the amount of pressure. Say you need a fight or flight response. you can constrict muscles in specific places to limit the amount of blood that goes to the liver if you need that blood to go to the skeletal muscle and so forth. I think my physiology is not that great, but there is also a tactical advantage to having higher blood pressure. I'd assume something about getting higher throughput through the system. Certainly there's problems with static high blood pressure, but one of your exercises talks about edema, where there is an imbalance between the pumping and your heart, and I think I'll have you guys break out today and discuss what those actual results are. But do pay attention to that one. It's a good thought exercise for do you actually understand where the blood's going in the system at a given time. I'll point out, one other thing. So that the capillaries, the very finest types of vessels, don't have this layer of muscle pretty much at all. So those would look a little bit different. They would have a structural difference. But odds are we're going to show you a picture like this that says, is it going to be an artery or is it going to be a vein? I will not really go through all the names of everything today because I can read a list just the same as you can. I'll point out a figure like this, that a lot of the structures, a lot of the blood vessels come in pairs. So if you have an artery sending blood to one place, you have a vein returning the blood back. And that's, look for those pairs. They're often named the same, and they should help you identify where you're at variously. This is your figure from your book. I would recommend knowing just about everything. Sorry. But this is your best summary for what are the different vessels that you need to know. If anybody comes up with a great mnemonic for, say, going from anterior to posterior, I will again promise you candy if you can share it with your colleagues, but of course it's important for your learning too. We will go through a few pictures of these things. We'll talk about putting some of the names to their functions, but again, that's something you should take care of, ticking off each item as you go through your pig today. What does each of these different arteries and veins service? So again, the different things come in pairs, so if you compare the vein figure with the artery figure, most of them correspond. Okay, so as you crack your pig open today, try to generate something like this. So I mentioned you should be able to take out the lungs. That'll give you much better access to the back side of the heart. I believe this also has a good chunk of the ribs cut out too. So today we're going to do some of our heavier butchering of our pigs, okay? Notice that you have access again to the backside, but that's where you're going to see these special fetal structures, including the Ductus Arteriosus. Try to identify this before any attempts to cut out the heart, to take a look at that in any greater detail. You're going to have to clear away a lot of connective tissue to get to the various carotids, jugulars, subclavians, it's ones that service underneath the clavicle obviously, but the carotids and the jugulars going to the head. Make sure you can find all the external and internal ones. Each copy, make sure you know about what distance is which so you could identify the difference given a practical examination. I may have mentioned before, please don't get the aorta wrong. It's huge, but I will admit that it's a little bit tricky looking at that and trying to separate it from, say, the esophagus. There's a lot of grayish tubes running around back here. So if you have a question, please ask in the course of the meeting. So that's another view, and I don't think I can add too much by talking about it again. There's a lot of vascular associated with kidneys. We will deal with them specifically in the urogenital section and excretory. Know now that they're a major filtering center, so that we need lots of vasculature to pump all of our different blood through there to be filtered. That filtration is not only about detoxification, it's also about homeostasis. How watery are you? How salty are you? But make sure you can identify all the veins and all the arteries coming to and fro. Once you're actually in the belly, make sure you can identify the major vein as well as the aorta. Note the vessels serving the genitals, and those are going to be finer structures. See here. Hopefully we'll have our very finely dissected male pig coming up next week, and you can see all these different things. But take care of dissecting away your connective tissue, your fascia, so that you don't disrupt those guys. Try to make small clips. But also make sure that you can identify the bladder in between the big umbilical vein and two umbilical arteries. As you get down to the pelvic region, we start calling things iliac instead of subscapular or subclavicle or jugular. Make sure you get all the branches, internal and external of these, and be able to distinguish between iliac and femoral. Okay, so looking at the test, these fetal structures are a big deal. I'm going to say it's probably 80% of what you'll get covered on this section, and there's a lot of stuff on vasculature. Okay? If that doesn't change in the meantime, what you need to know is the name of these three things, what they connect, and what their purpose is in a fetal pig compared to a normal pig. So those three things are the ductus venosus, foramen ovale, and the ductus arteriosus. Their names are somewhat instructive. You'll find that anytime you have a hole in anatomy, it's called a foramen, such as the hole in your skull that your spinal cord will go through is a foramen. So foramen ovale, start there, is just a hole, and it is a hole between the left and the right atria. It's actually a hole in the heart. Why on earth would you punch a hole through your heart to go from the left atrium to the right atrium? I'm going to pause this. Let's talk. So why on earth would you have a hole between the two atria? What are you skipping? You're skipping the lungs. You're pumping the blood directly from right atria to left atria so it can be loaded into the heart. The heart's still functioning, but the lungs are not, so there's no need to spend the effort to send them to the lungs where they will not gain oxygen anyway. Okay, that's foramen ovale. Ductus arteriosus is also associated with the heart. And this one I need to remind myself. Okay, so after class helps out Dr. Burns, we learn that the ductus arteriosus connects the pulmonary trunk, which is the pulmonary artery, to the aorta directly, thus bypassing the lungs. So there are two things helping us skip the lungs. Why do we have two things helping us skip the lungs? Because ultimately, neither one does the job completely. Some blood gets loaded into the right ventricle, and the heart is therefore trying to pump that through the lungs. We still don't want to do that. So that little bit ends up getting squirted through the ductus arteriosus directly to the aorta. So there's two holes in the aorta too. So there's some pressure issues there, but whatever. Ductus venosus is completely separate. Ductus venosus is down here. This structure is intended to help us skip the liver altogether. Liver doesn't need all that rich oxygenated blood, nutrient blood, but the brain, way downstream, really does. So ultimately, we pump this into the backside of the vascular system into the vena cava, where the blood mixes. So most of the fetus is never actually dealing with fully oxygenated blood. It's dealing with a mix of very deoxygenated infused with freshly oxygenated stuff coming from the placenta. Me and the colleagues had a good look at this figure because we were kind of obsessing over which is oxygen poor, which is oxygen rich, and we think there's an error in this. And that error is right here in the hepatic portal vein system. So normally in a fully functioning adult, you have a bunch of vasculature in your intestines. That's why when you eat, all the blood rushes down there to help absorb the food and you get sleepy. It's not because of the tryptophan in your turkey, it's because you got too much blood flow in other places. From the intestines, it goes up through the portal system into the liver, where we have the first steps in storing that energy, storing it as glycogen and so forth. So normally this is a really rich source of nutrients in a fully functioning critter. Here, but that doesn't have much to do with oxygen, I'm sorry, but both travel the same in the fetus. Here they show this is very rich in oxygen despite flow coming from the intestines, nowhere near a source of oxygen, and meeting up with this system where we're trying to skip the liver. So no part of this makes sense because they say the flow is in this direction and the source of freshly oxygenated blood is here. So apart from that, I think you can pay attention to the color coding on this figure to help you figure out which ones actually have the oxygen and which do not, which is important, which we will test you on the matrix of all the combinations of that. So please study and understand which structures are oxygenated. I have listed some certainties over here. Please take a look at those on your spare time. This is just another nice figure, and I left it in there for your reference, especially if you're working off of the PowerPoint itself. But I will use it as an opportunity just to reiterate. Remember, when you're a fetus, you're living off of your placenta. That's the interface of the fetus's vascular system with mom's, and that's where all the gas exchange, waste exchange is happening. So communication through the umbilical cord and through the vasculature associated with it, the umbilical vein, which is the big one coming in with the good stuff, and the umbilical arteries, which are the smaller ones wrapped around it, carrying the deoxygenated blood back to the placenta to receive its oxygen. So please be aware of that system too. That's it. Let's get cutting.